Hello everybody, my name is Tatiana Hernandez and I'll be presenting on Hepatitis A for Dr. Cody's disease report. So what is Hepatitis A? So some general information from Hepatitis A is that it is a hepatitis, which means that it causes inflammation in the liver, and that is in the genus. So let's backtrack a little bit on the taxonomy of Hepatitis A. So it's part of group 4, and if we remember that group 4 viruses are single-stranded RNA viruses, and in this case it's a positive single-stranded RNA virus. And it moves to the order picoRNA viralis into the family picoRNA viridae, and then it goes to hepatovirus. So a kind of interesting thing when it comes to the order and family, we have three kind of root words in here. We have pico, we have RNA, and we have viralis. So viralis is obviously talking that it is a virus. Pico means that it's a very small virus. It's between 10 to 30 nanometers. And then RNA is just that indication that it is RNA dependent when it comes to spreading its genetic information. So I kind of found that pretty interesting that the order and family in its own tells you a little more detail about the structure of the virus. The actual shape of the virus is an icosahedral, and here's an image of how possibly it sort of looks like. And you can see that it has 3 protomer sections. That's a little bit more detail on like how it eventually will present itself in the cell, but just as in generality, that's the shape of a hepatitis A virus. And we can see that there's all different types of species of hepatitis. Sometimes it's congruent to be called a hepatovirus A, but for most layman terms, it is called hepatitis A. And we have B, C, D, E, and then recently there's been a disturbance of another hepatitis, and it's called not A, not B, because it presents itself sort of similar to those two, but it's not those two. That's under the investigations. Hepatitis A is the most acute one out of all of them, meaning that it doesn't cause as a high rate of death as the other ones, and it doesn't persist as long or cause chronic signs and symptoms. So a little background about hepatitis A. In general, hepatitis is known as the oldest disease known to humankind according to the World Health Organization, and it hadn't been individually discovered to have different types of species. We knew that it was hepatitis, but we didn't know that hepatitis had different forms of itself. And it was kind of hard to be calling the hepatitis. Usually it was associated with something else, because the signs and symptoms just didn't really make it obvious that it was a different type of virus. It most assumed that it was associated with another type of illness. But in 1969, we have Dr. Baruch Bloomberg, and he was trying to do some hemophiliac studies. And what he was doing was trying to understand if the viruses and diseases could be transferred through genetic material, so from parents to their children and so forth. So was it more prominent to a certain family, or was it actually something random that you could catch from the environment? That was his study, and he was working a lot with blood transfusion patients, because that would have been the most obvious way to show that there was an issue with blood transferring to new people and they would catch the virus or disease. And then he kind of was doing more of those ELISA bloods and just trying to count antibodies to his certain patients. And when he went to Australia, one of his locations of studies, he found a new type of antibody that was prominent in a lot of people in that area. And he was first to discover that there was a disease called hepatitis. And what he presented it with was mostly it attacked the liver, which where the hepat comes from, and the antibody count increased for those type of liver cells. So he was the first one to discover hepatitis. And then in 1973, we have Stephen M. Feinstone, who eventually discovers individually that there is a hepatitis A, and that there is a wide variety of different types of hepatitis. In 1969, when hepatitis was discovered, the actual hepatitis that was discovered was hepatitis B, a more chronic hepatitis, but we later found out that there's all sorts of different types of hepatitis. So how do you catch hepatitis, or how are you exposed to it? And the first thing is to know that it's a fecal oral route. So it has to actually make it within your pharynx or through your intestines, which would have been like from your anus. And the reason that it's because it actually goes into your epithelial skin cells, and that's the only way it can actually first reach there. And it goes through your blood system to eventually reach your liver. So the most common way, is to actually get it foodborne, and it is through contamination of food. Here we have an examples of shellfish. Shellfish is the most common way to get contaminated with hepatitis, because shellfish is a good pool of where hepatitis could live. But you can get it from other foods too if it's not handled properly at a restaurant, not cleaned properly, or it's just there's already infection in the food you're consuming and it's not cooked properly. And what makes hepatitis so prominent, because it is prominent, is that it's actually resistant to many things that we don't expect, at least like bacteria to survive, and some viruses. It actually resists detergents, which would have affected the structure, and as well it would affect it's resistant to acidic conditions, so it can live in low pHs. It can actually live in high temperatures. It lives up to 60 degrees Celsius, and it's also resistant to other types of chemicals like ethers and chloroforms. So it's a lot of different chemicals and environments that it can actually live. And it actually lives for a really long time. It's actually lives for months in fresh water and salt water. So it just shows you that the condition of salt concentration doesn't affect it. And another key thing about hepatitis I forgot to mention, it's an unenveloped virus. So I guess that kind of shows you that there's a lot of things that don't really destroy hepatitis, and that's why it's able to live for a really long time. Now we can go to the pathogenesis of hepatitis. So as I mentioned, you consume it, or it gets in your system. It goes through your epithelial tissues, most likely in the pharynx and the intestines, and then it attacks your liver, going through your blood system. And the sites, you could see the counts of hepatitis by the antibodies in your hepatocytes and your Kupffer cells, which Kupffer cells are another type of liver cell that is part of your immune system. And it goes in through endocytosis. So I have images here. The image down here is a good example of endocytosis. This is hepatitis A. It's a little bit more complex. So I like this image here, that it shows once it's endocytosed, the single-stranded positive RNA goes into its double-stranded RNA, and then it's able to duplicate, and then it's released. And the reason it can do this is because the receptors of the hepatocytes are in match with the hepatitis virus, and that is easily recognizable, so it easily replicates without having any problems. It does get flushed out eventually, so in weeks or a couple of months it does get flushed out completely, but it does replicate really fast, and it replicates in the cytoplasm. So signs and symptoms of hepatitis includes jaundice, which is yellow skin and yellow eyes. So you can see here in this person that the eyes are really yellow where it's supposed to be really white. And it's kind of hard to notice yellow skin because, well, you have to know how they looked like before they had the yellow in the skin. Most patients will tell you the skin looks yellow. So here's a good picture of a yellowed skin. There's also pain in your stomach, or they said the side pain in your belly because that's how most patients would say it. Loss of appetite, nausea, fever, diarrhea, and fatigue. So basically just a lot of discomfort is the main thing about hepatitis. How is it diagnosed? Well, as I mentioned, it's actually detected through the antibodies that are produced in the body, and there's specific anti-hepatitis antibodies that are presented. So an ELISA blot is an easy way to detect it. And here's like a quick image of an ELISA blot. So you have your virus, you have your primary antibody, and then you add a secondary antibody and another detector, and then it changes color, and then based on, sometimes you detect by concentration, other times you just detect if it's present or not. So this is just an example. It's not exactly how hepatitis A is discovered, but that is just a little brief overview of an ELISA. And as you can see on this diagram to the right, we are able to see that there's immunoglobulin M and then immunoglobulin G. And immunoglobulin M is the most prominent one in the body. That's the first response So there is an increase, and within the first 2 weeks is when we're able to finally detect the hepatitis. And then we go to immunoglobulin G, which is present to patients who have been vaccinated for hepatitis. And after 14 weeks, the hepatitis is gone. Another detector is alanine transferase, which is another immuno response from the liver, and this is specifically cells for the liver. So that has an increased spike, and that's how it's also detected that it's hepatitis. So there are vaccinations. Vaccinations did start in 1969 with the discovery of hepatitis B. It was considered anti-cancer because it actually helped prevent some liver cancer, because without that immunocompromise from hepatitis, you had a lowering of your chances of getting cancer. If you are exposed to hepatitis, you just get more immunoglobulins to help you out. But the most common right now is this Twinrix vaccination, discovered in 2001, but it has hepatitis vaccinations have been given to patients since 1990. And it's a 2-dosage vaccination to increase your immune response, given to young children, and it's recommended to every child in the United States. It's kind of required in a lot of school districts. So you might have heard hepatitis through that, because you more than likely do have the vaccination for hepatitis. And a couple trends about hepatitis: it's more likely to be seen in developing countries due to socioeconomic factors, cleanliness of the environment, and getting clean water, as well as some cultures, depending, because some cultures eat certain foods that would make it more likely for you to be infected. Eating raw fish, for example, is an example for countries that are more prominent to do that. It's more likely to get hepatitis. And as you can see in Europe, Canada, and the United States, it's not very likely. And there has been a few outbreaks in a couple of years here in the United States, and it's usually due to food contamination, and it's not always meats. It's sometimes in these 2003 Ohio and Pennsylvania outbreak, it was actually detected back to unclean green onions. So hepatitis can attach itself to anything. But as you can see in this trend right here, it's actually been decreasing, and it's lower and lower, and it doesn't really depend on age group. It's kind of the similar trend on everybody. So here are my references, and I'm glad you listened to my presentation. If you have any more questions, just ask me next Wednesday.